Today's lecture is gene testing. Gene testing is also called DNA testing. First one is predictive testing. Tells a person if she carries a mutation that will cause or put her at higher risk for a disease later in life. Next is newborn screening. Detects common disorder in newborns where immediate treatment can prevent dangerous symptoms. Now, career testing tells a person whether or not her carries a mutation that could be passed on his offspring. One can be career but not be a risk for a disease. Career testing is just like as in recessive gene. When we are affected by any genetic disease, what does a genetic counselor do? He also gets the testing family members and with disease symptoms to find out about disease. Support is needed for individuals who need to consider issues such as stigma disclosure to family members and confidently. Counseling to reduce potential psychological distress. Counseling should be available to provide information about genetic risk and explain choice regarding genetic testing and further management. Patients need to sufficiently informed about the implication of genetic screening before they can provide. There is often little you can do to prevent a genetic disorder, but genetic counseling and testing can help you learn more about your risk. It can also let you know the likelihood of passing some disorder on your children. The pathway from DNA to protein, the flow of genetic information from the DNA to RNA transcription and from RNA to protein translation occur in all living cells. Now, levels of genetic testing. Analysis of whole chromosomes. For large changes, extra chromosome, very large deletion or insertion. Analysis of sequence. For small changes, mutation in the sequence, small deletion or insertions. Analysis of protein safe for any change that may affect folding of the protein. Analysis of protein function. If the functional protein is supposed to make something, then some test can detect the presence or absence of the product. So, how do we look at the DNA sequence? This procedure is done by sequencing, RFLP analysis, probes, dot plot, microarray, gel electrophoresis, PCR. Let's talk about gel electrophoresis. By this technique, separates DNA fragments on the basis of size. Large fragments take longer than small fragments to migrate through an agarose. The next technique is PCR. PCR, polymerase chain reaction, is the process in which multiple copies of gene or segment of DNA of interest are synthesized in vitro using cDNA and DNA polymerase. Polymerase chain reaction was first developed by Carey Mullis. It is directed by a machine called thermal cycler. A standard polymerase chain reaction setup consists of 4 steps. First is add required reagent or master mix and template to PCR tubes and mix and centrifuge. Then amplifier per thermocycler and primer parameters. Evaluate amplified DNA by agarose gel. Electrophoresis followed by ethidium bromide staining. A single PCR amplification cycle involves 3 basic steps: denaturation, annealing, and extension. In denaturation step, DNA is heated to 94 degrees Celsius to separate the two strands. The second is DNA is quickly cooled to 30 to 65 degrees Celsius to allow short single-stranded primers to anneal to their complementary sequence. The optimum temperature for this extension step is 72 degrees Celsius. The entire cycle is repeated. How do we look at the DNA sequence in RULP analysis? Restriction fragment length polymorphism analysis relies on the uses of restriction enzymes. These enzymes recognize specific DNA sequence using palindromes and cut them. RFLP analysis means that there are polymorphism between people in the number of restriction sites. This person has four fragments. This person has mutation that eliminates on the sites that the restriction enzyme cuts at. Therefore, this person has three events, one of them being much larger than the rest. If this mutation was associated with a disease, a restriction digest would show that carrier of the mutation had three fragments instead of four. Issues with RFLP analysis can't detect all mutations. The mutation has to coincide with the restriction enzyme cut site. Now, sequencing. Here we have known gene sequence. How do we find out if the patient sequence is different? How do we look at the DNA sequence in probes? There are DNA bases paired with complementary bases. A probe is a short fragment of DNA that is complementary to part of a longer DNA sequence. DNA strands can be separated with high heat. As the temperature is lowered, smaller fragments that have complementary sequence will base pair faster than the long original strands of DNA. The ability of probes to bind depends on its complementary to the DNA strands. Its binding to single base pair differences can affect binding. The stringency of the binding conditions depending on temperature buffer conditions. We can see in this figure low stringency, high stringency. Membrane is washed in a solution that contains radioactively labeled probe. Probe bind to complementary sequence. The next technique is microarray. In microarray, if a patient's DNA bind to spotted probes, a computer detects this by measuring the intensity of the fluorescence emanating from the spot. A homozygote for a CF mutation will have a more intense spot than a heterozygote for the same mutation. Benefits of genetic testing. If negative, relief, fewer healthy checkups and tests that go with being a family and tends to have higher risk of particular genetic disease. If positive, able to make informed decisions may be possible to reduce the risk coming down with severe symptoms. Limitations of genetic testing. First is mutations may not always lead to disease. Exiting test only for the most common mutations. Some disease causing mutations will not be detected by conventional tests. Small chances of errors in testing procedure. Testing is not always matched by treatment. The last slide is where do we go now? Try to resolve ethical, legal, social issues. Expert in science, ethical law, policymakers. Develop new, cheap, simple, effective, and accurate tests. Testing for all. Advanced research in disease treatment, gene therapy, and drug treatment.